Pancreas is derived from the endoderm of the embryo. The pancreas lies inferior to the stomach in a bend of the duodenum. It is both an exocrine and an endocrine gland, i.e. heterocrine gland. A large pancreatic duct runs through the gland, carrying enzymes and other exocrine digestive secretions from the pancreatic acinar cells to the small intestine. The tissue of the pancreas has, in addition to the acinar cells, groups of cells called islets of Langerhans. Types of cells identified in the islets: 1. Alpha cells, about 20%; 2. Beta cells, about 70%; 3. Delta cells or D cells, about 5%; 4. PP cells or gamma cells, 5%.
पहले मैथ्स या साइंस या ज्योग्राफी, पढ़ाई से ज्यादा पढ़ाई के प्लानिंग में टाइम जाता।
1. Glucagon, secreted by alpha cells. It stimulates the liver to convert stored glycogen into glucose. Glucagon is controlled by feedback in accordance with the level of glucose in the blood. When blood sugar rises, secretion of the glucagon is suppressed, and when it drops, secretion of the glucagon is stimulated. Target cells. Glucagon acts on the cells of the liver and adipose tissue. 2. Insulin, secreted by beta cells. It was first found by Banting and Best. A.F. Sanger coined the term insulin and proposed the molecular structure of insulin, cow's insulin. Insulin was the first protein that was artificially synthesized in laboratory and crystallized. One molecule of insulin is made up of 51 amino acids that has two chains. I, alpha chain, it is made up of 21 amino acids. E, beta chain, it is made up of 30 amino acids. Both the branches or chains are bind together with cross bonds of disulfide bonds. Functions. I, it is antagonistic to glucagon. Insulin decreases the level of glucose in the blood. It acts by increasing the rate at which glucose is transported out of the blood and into cells, and by stimulating muscle cells to take up sugar from the blood and convert it to glycogen. Like glucagon, insulin is primarily regulated by feedback from the blood glucose concentration. When blood sugar level drops, the secretion of insulin is suppressed. When blood sugar level increases, the secretion of insulin is stimulated. E, it promotes protein synthesis in tissue from amino acids. E, it reduces catabolism of proteins. It is an anabolic hormone. IV, it increases synthesis of fat in the adipose tissue from fatty acids. V, insulin reduces breakdown and oxidation of fat. 3. Somatostatin, SS, it regulates secretion of insulin and glucagon. Target cells. Both somatostatin and pancreatic polypeptide act on the cells of the pancreas. 4. Pancreatic polypeptide PP. It appears that pancreatic polypeptide inhibits the release of digestive secretion of the pancreas. Both somatostatin and pancreatic polypeptide are relatively newly discovered hormones of the pancreas, and both are still being studied.
Guess what I'm super thrilled about?
Diabetes mellitus hypoinsulinism. The insulin-dependent diabetes mellitus (IDDM) is caused by a failure of the beta cells to produce adequate amounts of insulin, while non-insulin-dependent diabetes mellitus (NIDDM) appears to involve failure of insulin to facilitate the movement of glucose into cells. 1. Hypoinsulinism, hyperglycemia. 2. Hyperinsulinism, hypoglycemia. Pineal gland, epiphysis cerebri. It develops from the ectoderm of the embryo. The pineal gland is located between cerebral hemispheres, where it protrudes from the roof of third ventricle. Pineal gland is a small rounded body which consists of I pineal cells, E, supporting glial cells. Hormone. 1. Melatonin. 2. Serotonin. Thymus gland is derived from endoderm of the embryo. Thymus